Yes, yes, we can see full screen.
Okay. Yeah. All right. So this is an area of interest for me after doing my PhD in immunology and having loved physics and math all through college. We became very interested in oxidative stress as an inducer of lung injury. And after studying another chronic lung disease in premature infants that's caused by oxygen, I was questioning whether the same mechanism could apply to radiation-induced pulmonary fibrosis. And interestingly, the people who treat patients with this illness, it never goes away. It very slowly exacerbates to a condition similar to idiopathic pulmonary fibrosis. But I see it as a model of what might happen in those patients with IPF, which is idiopathic pulmonary fibrosis, in the early stages where you can't really see what's going on. Patients don't have symptoms. This is a very, a very slow process. And now how do I get to the next slide? I just go, oh, there. Oh, this is just for the record, I always have to have a disclosure slide. And I've been at Duke for 17 years now. And I have previously been funded by the NIH as a PI, as well as a co-investigator on, and I've had two different pilot projects on the group radiation countermeasures grant in 2007 and in 2015 with Mark Dewhurst and then Nelson Chow. So this is what got me very interested in this area. The peptide we've been studying is a little unique. It is only a 14 amino acid bioactive peptide that was first discovered in the skin of an amphibian. It's not a frog, it's really a toad, but the name of the frog is Bombina bombina. So the name of the peptide became Bombesin. And the terminal methionine has to be cleaved and amidated in order to be active at the receptor. Using antibodies to bombesin, a number of scientists began looking at mammalian tissues, and they identified again, a 10 to 14 amino acid peptide derived. The 10 amino acid is just a cleaved product of the 27 amino acid mature peptide. And interestingly, the highest levels of this GRP, as we call it, are in mid-gestation human fetal lung. So we spent about 15 years looking at how GRP can promote fetal lung development with branching morphogenesis and cell proliferation and differentiation. And the cells that it's made by are also very unique in that they are the oxygen sensing epithelial cells that have many similarities to neurons. And therefore they store these peptides in dense core neurosecretory granules, just like neurons. And there are two main receptors, the high affinity GRP receptor and a low affinity related neuromutant B receptor. So this took a lot of our focus for the first while. And then we started looking at chronic lung diseases. Initially, hyperplasia of the neuroendocrine cells was observed in premature infants that survived a week or more in the 1980s. And this was in contrast to immediately after birth, the number of stainable neuroendocrine cells was decreased. So if you see this by fluorescence, it looks like a starry sky because these are just like little nodules of cells scattered at branch points throughout the lung. And this peptide can activate macrophages, including phagocytosis, cytokine secretion, and cell macrophage migration, as well as fibroblast proliferation, migration, and differentiation. And okay, so I like this because it shows where we are at the moment with the animal models. We've been working with a C57LJ mouse, which is highly radiation sensitive. And we've been using X-rays sort of in the middle range of the X-ray spectrum. Although if it gets to the point of looking outside the Earth's orbit, we're going to have to try to apply the same principles, looking at gamma radiation. And I think a lot of people don't realize how protected everybody is due to the Earth's atmosphere. And so our hypothesis is that if this is an airway epithelium in the lung, these are club cells that at a branch point in the airway, you find these clusters of neuroendocrine cells, which will degranulate mostly out in the basolateral space, but some of it does leak out into the airway itself. Whether these cells are triggered by radiation, which is the topic of today's talk, or other reactive oxygen species such as hyperoxia, also hypoxia, because in the case of hypoxia, reactive oxygen species are generated as well. Also ozone and on and on. So GRP, I made these cells green because GRP was found in a frog. And so you can always remember that. And here we have, immediately after being released, GRP will draw in the neutrophils, which have chemotactic activity and are also, they marginate through the endothelial cell layer. But endothelial cells also have GRP receptors and macrophages. The macrophages here is like, and CD4 positive T cells and macrophages have the highest levels of GRP receptor, as well as the neuromedin B receptor of any cells that we've looked at. And later, this is a very early event after radiation, but fibroblasts become activated and differentiate into myofibroblasts, which are a very, it's not super early, but it's one of the earliest stages of overt collagen production towards fibrosis. So these will start to migrate and proliferate and infiltrate through the endothelium. They can go through the endothelium, or they can simply migrate from other parts of the lung, such as in the perivascular and the airway interstitium. And these cells will migrate, proliferate, and also differentiate into the next level of fibroblasts are synthetic fibroblasts that produce predominantly elastin and collagen, which are an important part of the basic lung structure, but at the wrong place in the wrong time will cause organ failure and death, because you won't be able to exchange oxygen if the interstitium is taken over by connective tissue. And at the time that these are produced in large quantities, the myofibroblasts have essentially disappeared. So you just end up with an end-stage lung. So here's the mouse model that we published in 2019. And as you can see here, we've been, in order to look at urine levels of GRP, which have been used in studies of lung cancer patients, as well as we managed to identify in premature infants that a GRP in their urine in the first week after birth predicted whether they would get chronic lung disease months to a year later. So it is quite predominantly detectable in the urine and not detectable in the blood because there are binding proteins and it's just not measurable. So we used metabolic cages to collect urine in 6-hour intervals from within 10 minutes after radiation to 6 hours, then to 12, 18, and 24 hours. And when you, how am I gonna get myself out of this? I'm sorry. So the percent change in GRP is shown here as the negative controls that were not irradiated. In each experiment, the 0 to 6 hour window shows the radiation treated, mice treated with radiation and PBS, or radiation then treated with a very high affinity neutralizing antibody to GRP. And it's actually interesting at this early stage that there's a little bit more of the GRP detected in the urine, which could be due to enhanced clearance. But the GRP in the urine over that full 24 hours stays pretty much elevated, and it starts to come down a little bit, but it's still about significantly above the baseline value. And the animals that got the antibody have a significantly lower GRP than those that did not. And this is the reference to the paper. And interestingly, although this peptide is so strikingly elevated, we looked at two different full experiments with a multiplex for cytokines and inflammation and fibrosis, using lung at 24 hours after radiation. And the only one that was about 30% increased in radiation with radiation alone was IL-10, which is a funny cytokine that can have a role depending on the situation in either fibrosis or in protecting against fibrosis. But you can see that with the antibody, this came right down to baseline. And when we took lungs from mice 6 hours after radiation, you can see when you stain for GRP with the same antibody, 2A11, that most of the GRP is in blood vessels. So this is a vein, and here's another vein. And then also lining all the alveolar spaces, indicating that, and this is at very high power, you can see that this is in the lumen. It's not on top of the cells. And here's another one in the lumen. And if you take the antibody and totally absorb it onto plastic polystyrene that will be coated with GRP itself, And then you incubate the antibody with this solid phase that has been washed extensively. You get much decreased staining with the antibody, showing that all of these localizations are basically GRP. And that's, and we did phosphoproteomics, which is the beginning of probably a lot more omics if we wanted to do a time course. And I'll just show you the difference between the fold change in these various gene expressed genes, comparing antibody treated versus radiation alone. I mean, they're all radiation treated, but it shows the change with the antibody. And you can see there's a threefold increase of genes that are found to be decreased with ventilator-induced lung injury and edema. And that also reflects a functional capacity of the endothelial cells. The ATP binding cassettes are overexpressed in cells that are resistant to ER stress. Protocoherin binds SMAD, which is a downstream signaling molecule on the TGF-beta1 pathway. So if protocoherin goes down, this would be anti-fibrotic, and also it goes down, and protocoherin goes down in patients with smoking who have more GRP. Then others, there's a couple more. There's one septin-4, when it's missing, the mice are predisposed to hepatic fibrosis. Spark-like protein promotes endothelial cell quiescence. And then there are a lot of molecules I mean, some of these, the names are so long, I didn't write them here, but the one heat shock protein that predicts a poor outcome in sepsis goes down. So this is it's like an anti-idiotype. You have a low level of a heat shock protein, whereas the high levels of the same heat shock protein predicts a poor outcome. So it's an anti-anti. And also other things, the PEAK kinase that promotes it promotes epithelial mesenchymal transition, which is part of fibrosis. Okay, and then here's the mice again. And you can see we treated them with 15 gray and then injected the antibody at 24 hours after radiation, because the NIH is only interested in drugs that you could give people if there were a mass casualty radiation accident at a time afterwards. And now alpha smooth muscle actin is a marker for those myofibroblasts, but it's also in normal smooth muscles. So in mice that were not exposed to radiation, you can see it's in smooth muscle around airways and blood vessels, but not out in the periphery. After radiation alone, a lot of the small blood vessels and even capillaries have staining for myofibroblasts, meaning that they're creeping out to the periphery and beginning to interfere with the air exchange. And if the mice are treated with 2A11 anti-GRP 24 hours after radiation, and then you look at, actually, I aired here, this should actually be 10 gray for this experiment. So, But anyway, you see very little out in the periphery. Maybe, I think there's one tiny little spot in here. And trichrome is a histochemical stain for collagen. And we actually have done this in parallel with staining for collagen itself. And it exactly matches the distribution of collagen. So normally you only get collagen around airways and blood vessels. In the mice with radiation, it's out all through the periphery, and it's green. So it's a little bit hard to see in this picture, but you can see it's actually bridging a whole area of the distal lung. And with the antibody, there was very little identified. We did image analysis with an image J 1.62 that's available from the NIH, and it's a freebie for anybody who wants to do image analysis. So we can set this to only detect green wavelengths like this, and then we normalize for the numbers of nuclei present as a rough estimate of the area of tissue. And the counter stain is hematoxylin, which stains purple. So it's a totally separate wavelength from the green. And this shows the percent immunopositive alveolar tissue area, which you can see in normal mice. There's myofibroblasts and collagen. These go up. This actually goes up 18-fold for SMA. And collagen goes up, this is more like 1. So this one goes up about 5 to 6-fold. And then with the antibody treatment, these come way back down. They're not completely normal, but they're pretty low. And those are all highly significant. So we'd also been working with a non-human primate rhesus monkey model of radiation-induced pulmonary fibrosis, along with Mark Klein and Priyanka Thakur at Wake Forest University. And the animals are sedated, treated with 10 gray of just thoracic radiation. And then we give them at 24 hours a single dose. We were limited in the amount of antibody that we had because of the size of the animals. So they got a lower injection than we had been giving to the mice. And then we only did trichrome staining at the 9-month survival point. And okay, so across here, all of the normal non-human primates. Now, there was one animal that was sick and we didn't realize it before it was irradiated. It had an extremely high eosinophil count and like a 3-fold increase of eosinophils and 1/3 as many lymphocytes. So this one animal died, but all the other animals that got the GRP antibody survived to 9 months, as opposed to about 59% of all the animals without the antibody. And this is not significant, but it's only 9 radiation alone and 3 radiation with 2A11. And so when you analyze the slides, you can see a normal non-human primate. There's a little bit of blue here and there because the airways cross over, and you can see that like in respiratory ducts. So here is with radiation alone, and there would be widespread pockets of collagen, and there'd be some other areas that didn't look so bad, but we had about 6 slides from each animal, and we I'm a nitpicker for making sure I believe my own data. So we take the slides and we tape the label side twice and give them to an administrator to write down what the slide said and what number it has. So it'll be like, we had like 30 slides from all the animals, and did this double blind. And then when we matched them all up, each individual animal could have an area that didn't look so bad, but because of the beam. It depends where the slide came from, you know, whether it was in the middle of the beam or to the periphery. But when all the numbers came together, you can see this is on average what, with the 2A11 antibody, there's very little fibrosis, and it's, these are considered nodules that sometimes bridge a little bit, but it's, you don't see anything like these big areas of infiltration and nodules. And so if you look at the modified score, there's an objective score by a, I think a German histopathologist, which is very easy to score. And with the antibody, the scoring of the lungs goes down about half. And so we still have slides to do, but it's the beginning. And now this is not going over. Let me just see where it is. It's not going. Okay. Okay, let me just try to get this out of the way here. Okay, so I can just stretch this out here a little. So there we go. Oh, here, I just use that one. So I wanted to summarize the major observations that in mice and non-human primates, neuroendocrine cells degranulate in response to oxidative stress, including hyperoxia, hypoxia, ozone, and importantly, especially for this conference, ionizing radiation. And in the lung, ERP is secreted within hours to days after exposure to radiation. And if you block TRP, you can prevent pulmonary fibrosis months later. Within, in addition, a recent paper of premature infants showed that urine GRP levels strongly correlate with oxidative stress levels in the same urine samples, but only the urine levels in the first week after birth. After that, they do not predict diagnosis. And is thinking about how these cells are similar to neurons and realizing that one of the obstacles for travel to Mars or even the moon or away from the protection of the, you know, the geophysical barrier around the Earth, that similar degranulation of neurons or a subset of neurons exposed to ionizing radiation that is known to lead to cognitive defects, deficits, could be irreversible. And so what I was thinking in terms of, you know, today's group of interests is that, okay, here we are, and we have geomagnetically trapped radiation that we can shield. But if you look at what comes to Mars and the moon, or these large solar proton flares, as well as cosmic rays. And this is what could be a new productive area of research to identify which neurons are most damaged by radiation. Like, what are the most radiation sensitive neurons that could be involved in cognition? And could you discover new therapies to maybe neutralize the toxins that might be involved to minimize brain damage during prolonged space travel? And oh, and this is just to acknowledge the people who've been instrumental with this. It's like a bunch of chair people, Dr. Pizzo at Duke, and then Dr. Cotran and Ben Asraf at Harvard and Brigham and Women's were my chairs, and my research mentor, Martin Dorff, as well as Jaco at University of Maryland now, Mark Dewhurst, Mark Klein, and Priyanka. And I thank you for your attention.